All right, topic 3.1, part 2, right? So in topic 1, we ended here looking at that definitions of genes, loci, and alleles, mutagens, types, make sure we know those things. And now in part 2, we're going to look at a genome, actually look at the comparisons between different species, and then using databases to identify genetic features. Ooh, mapping out the human genome. So let's clear this out. Oh, sorry, hold on, back. Oh, pen, clear, back, haha. Okay, all right, let's go back to where we left off. Here we go. Okay, so definition of a genome. We've actually already looked at this. Like, wait, the genome and the proteome, we talked about like why the proteome is larger than the genome, right? So different amounts of proteins can be made than the variety in a genome. Okay, so let's look at this. So genome describes the total genetic information of a cell or an organism. Right? Remember that a cell actually carries all the DNA. So when we're looking at the genome, we can actually look at a single cell because all of the DNA is in there, right? So it includes what we call the genes and the non-coding sequences. So if you go back to those chromosomes that we were looking at, the genes are the specific ones that code for a trait, general traits. Not all of them are coding sequences. They're called kind of like loosely like junk DNA. They're non-coding. They don't code for anything specifically. However, when we're mapping out the genome, that we include all of the all of the DNA, genes and non-coding. Okay. All right. So the genetic relationship between genes and genome, we're going to compare to the technology, right? So my genetics, right, all of those bases put together in certain orders is what's going to create genes. And then all of those genes come together to what we call the genome, right? So these are just A's and T's and G's and C's, but the specific order that they're in, right, is what we call, and the location, so order and location, right, is what creates a gene. And then all of those specific genes that we have together creates what's called the genome, right? So if you're in computer science, so you understand coding, right? Binary is just zeros and ones. So this is kind of like comparison to it, just zeros and ones. However, those zeros and ones come together to create different programs, right? So computer programming, and then all of those programs go together in the computer to make it function, right? So going back here, those codes, what it does comes together to make it function, right? So having some kind of order or analogy for how that works might be helpful as you move on deeper into this. Hold on. There you go. Deeper into this topic. Okay. So the Human Genome, or what is called as the Human Genome Project, was an international cooperative venture. So a lot of scientists got together all over the world with all their technology, and they're like, instead of like working in these silos, let's put everything we know together and work together to establish the sequence of the human genome, right? Let's map out the human genome. As you probably know, like some different organisms are more simple and they're easier to map out their genome. Humans very complicated, so this took a lot of energy, people, time, right? And all these different data sets coming together. So what we found from the HGP, you should know that kind of acronym there, is that humans share the majority of their sequences, right? So there's only a couple short nucleotide differences that is seen as contributing to all of the genetic diversity in mankind. So, right, like 99, I've actually the number is like 99.99% of the DNA is completely the same. Just a couple of those nucleotides in different places, different orders, creates the differences that we see in mankind, right? Knowing the numbers of that. So humans have roughly 3 billion base pairs in DNA in their genome. That's a lot of different pairs, right? This typically consists of 46 chromosomes. So those come together and make chromosomes. That's in our next topic. Typically because we can look at sometimes like Down syndrome, triple X syndrome, so different chromosomal abnormalities that still create or still create life, like people can still live, but usually cannot pass on. And the genome contains roughly 20,000 protein coding genes, right? So 20,000 protein coding genes. So 3 billion bases, 46 chromosomes, 20,000 protein coding genes. Okay, to look at that, right? So if I've got 3 billion and I only have 20,000 of those that code for it, a lot of them in between don't code, right? They're the non-coding genes that we talked about. All right, there we go. Okay. Genetics comparison. So when we're looking at different or comparing the genetics between, well, whether it's different animals or different between animals to viruses, viruses to prokaryotes, right? We're looking at comparing their general genetics. I was going to say in general. I need to go to sleep. Genetics comparison. All right, so separate species have very distinct patterns in different numbers of ways. So this includes a couple different factors, right? So different species have these three difference. They have a different genome size, the number of chromosomes, and the number of genes. We've already actually looked at the number of chromosomes here a little bit, right? Like the fruit fly has this many, and a human has this many number of chromosomes. So that's how those DNA is organized. There is no clear correlation between those trends. I'll show you just here, hold on. Here, sorry, I wanted to bring up this chart before I say this sentence. So no clear correlation between these factors, looking at like whether the number of chromosomes affects like its complexity or the number of genes or the genome. So for instance, if you've seen this chart, I think we looked at it earlier, right? So a human we talked about has 46 chromosomes, but a dog has 78, right? And a fruit fly has 8, right? And so this number here, like a mouse has 40. So a mouse has more chromosomes than a human, but I'm sorry, has less chromosomes than a human, but has more than like a cat, right? So it doesn't has no correlation between like the number of chromosomes and its complexity, right? So just know that it's not like the smaller the chromosomes, like the less complex the organism is. Okay, but what we do know, there's some trends, right? So not correlations, but trends. So we do know that viruses and prokaryotes tend to have smaller genomes and fewer genes, right? Which would make sense, right? Because they're not as complex, they don't need as many proteins. So they tend to, but not always, have smaller genomes and fewer genes. Eukaryotes, so plants and animals, have a huge variation in both genome size and gene number. And that's what I just showed you. So from a dog to a cat to a mouse, right? To all those things, like there's a huge variation in how many chromosomes and the genome size, right? However, you should know, right? So without any like, when I was saying, it says like, what are the following is a true fact, right? No consistent or reliable pattern between the genetic factors and the relative complexity of the organism, which is what I just showed you in that last chart. Let's see. Okay. All right. Moving on. All right. Just a little, here you go, visual here. The one I just showed you, that was chromosomes. This is looking at the genome size, so the number of BP. Remember, the B pair equals base pairs. So anytime you see BP, you're looking at base pairs. And that it's not a valid indicator of genetic complexity, right? So this is the common name, this is the genome size, and this is the species, right? So where are we? So humans, we talked about over 3.2 billion. A canopy plant, 150, fruit fly, 130, right? So this is the bacteria, 4.6 million base pairs. So in E. coli, has more base pairs than a human, right? So definitely not looking at complexity, still trying to figure out why that might be, right? Like what the relationship is between complexity and species. All right, here's the chromosome numbers that I just showed you as well. Looking at the chromosome numbers, do you need to know these? No, you should need to know that humans have 46. And we'll be looking at haploid and diploid numbers soon. But just know that, again, no relationship between genetic complexity and species. Okay, and one more just for good measure. Here's the gene number, so the genome size. Right, looking at all those different sizes between the same method. The estimation. So actually, I skipped over this one. So the estimation of gene number. Actually vary significantly depending on the prediction method. I think your book talks about the prediction method. Let me check for sure. But just know as we go to estimate the number of genes, that depending on the model that we're using, the prediction model, we can actually get different estimates. So long story short, these are not facts, right? So these are estimates of the number of genes. They are not something that you need to memorize, just the trends. Okay. All right. Last slide here. So genetic database. So there's a lot of different genetic databases. There's actually some controversy over genetic databases. Maybe we'll look at that in like criminal justice. But anyways, there's a couple online that looks at the comparison of genetics, like looking at bases, polypeptides, positions, all that kind of stuff. The two that you need to know about are GenBank and Clustal Omega. So GenBank contains the collection of DNA and peptide sequences. Remember, peptide sequences are what? You should have just said proteins, right? So my polypeptide sequences are proteins, right? So looking at the difference, the collection between those two. The clustral omega, this is funny, is an alignment looking at a program for comparing just different sequences, right? So it's not looking at the collection of the two and comparing it. It's just looking at the different sequences. There's some genetic databases, not here, that actually kind of, this is what you call theory of knowledge, like going bigger picture, looking at creating genetic databases for all humans, so that if when crimes are committed, you can actually just go to the genetic database and compare the DNA. There's some controversy around that, as you can imagine. And maybe we'll look at an article or two of that in class. Okay, we are finished with part 2. Oh, look at those marks there. Okay, so in this last one, the things that you should be able to do, right, are right here. So what is a genome? What are the trends? What do we know for sure? What do we not know for sure? Chromosome numbers. When I'm looking at different species, right, the different, whether it's chromosome numbers, the base pairs, different amount of the proteins that are made, and then the use of the databases to identify genetic features. All right, that is it for part 1. In our next section, we are going to be moving on to chromosomes, right? So looking at how those genes are actually stored in the form of chromosomes. All right, you guys have a fabulous day, and I will see you soon.